This is my week 4 session with you, and this will be my last session. So I'll try to wind up things and take your doubts. So, in this, we'll be discussing about previous session doubts, and we'll also proceed with the week 4 topics, and we'll practice MCQs related to the week 4. So, in week 4, you have gone through the comparative genomics, genomic insights into evolution, in which you have read about the power of comparative genomic analysis. So, now let's start the session. So in the previous session, there were doubts related to the probes, that how they are attached to microarray, because as we have discussed a lot about microarrays, that how they are helpful in identification of certain genes or certain fragments to check the expression of certain genes that are present in the sample. But now the main question was that how are the probes designed and how they are attached to the surface of microarray. So the technique of attachment of these probes to the surface of microarray is referred to as immobilization, and these techniques are known as immobilization techniques for the microarray. So their success depends upon the chemistry which is used for this immobilization process. So here I have discussed about the two types of predominant methods that are generally used. So first method is the direct in situ synthesis of oligonucleotides on the chip surface by using photolithographic methods, in which generally with the help of light the probes are attached, and there is a principle of physical adsorption which is being which is undergoing, due to which these probes get attached, they get immobilized to the microarray surface. And the next method is the deposition methods. So here are few examples. So the probes can be attached either due to physical adsorption, or they can be covalent immobilization, or there can be streptavidin biotin immobilization, due to which probes get fixed to the surface of microarray, and hence we can proceed with this analysis. And in this, I have provided you the some of the other methods that are being used for this fabrication. So this immobilization is also referred to as fabrication. So this fabrication means that you are attaching the DNA or any oligonucleotide or RNA to the surface of the microarray. So that is known as DNA microarray fabrication. And for this, different mechanisms or methods which are used are robotic pin spotted microarray, in situ microarray in which the photolithographic method is used, then inkjet printing microarray, polymer photodeposition of microarray probe positions, and the last one is high density fiber optic microsphere based microarray. So these are different methods that are being used for this DNA microarray fabrication. And you need not to go into the depth because these are not in your syllabus, but this is just to give you the idea that how the probes are attached. Then here I am providing you with the table in which I have given you the list of probes which are generally used for the microarray, and here are the applications that for which analysis you select the particular probe. So here are few probes such as cDNA probes, oligonucleotide probes, protein probes, peptide probes, ligand probes, tissue lysates, nucleic acid aptamers, carbohydrate probes, DNA binding protein probes, and RNA probes. So these are some kind of probes that are generally used in the microarray. And here are few applications. So the applications are, for example, cDNA probes. So when we use them, when we want to see the gene expression, or we want to study the mRNA levels, and we want to detect the alternative splicing events. So these are few of the applications of cDNA probes. Similarly, oligonucleotide probes are used when we want to check gene expression analysis, genotyping, SNP detection, studying miRNA expression, and microRNA profiling. So similarly, you can go through the other probes, that when are they used and what are their applications. Then the next doubt was related to the table. So when we were looking into the multiple sequence alignment, then there we have observed that when we compare the protein sequences from multiple species, then the amino acids which are having similar biochemical functions, or which are not affecting the protein function or structure, they are referred to as conserved mutations, or we can say conservative substitution or conservative mutation. But in that table, one thing which was a little weird was that proline to glutamic acid mutations, they were referred to as conservative. So the question was that both of them shows entirely different biochemical properties, then how this mutation is referred to as conservative. because it appears that they might have drastic effect in the protein structure and function. So the table which I have presented in the previous session, that was obtained from a uh, published patent in which they have performed the analysis on a particular protein, which is tropoelastin, and I'll discuss it in detail in the next slide. So first of all, what was the reason that why are these mutations were still considered as conservative mutations? Although in that the level of conservation was not mentioned, so they were mentioned as conservative, but it doesn't mean that it is also as same conservative as the other mutations. So this mutation is less conservative as compared to the other conservative mutations. But why it was conservative? Because this substitution do not significantly affect the protein's function or structure, and if it provides some selective advantage to the organism carrying the mutation, then also this particular mutation is considered as conservative mutation. So these are few reasons that why we call some mutation to be a conservative mutation. So means instead of these having different biochemical properties and different structure, but still they are offering some kind of advantage to the organism. So that's why these are known as conservative mutations. And there are certain factors which govern that when can we call a particular mutation as a conservative or non-conservative. So first factor is functional tolerance, which means that some proteins can tolerate this proline to glutamic acid substitutions without a loss of function. So in those cases, we'll call it as the conservative mutation. The next is genetic redundancy, which means that the redundancy in the genetic code allows different codons for the same amino acid, which is facilitating the substitutions. So in this, they are also telling you the reasons that why you observe it as conserved. So conservative term means that particular mutation or substitution is seen in all. So whenever some mutation have some drastic effects, then it generally do not remain conserved. It get disappeared. So if something is conserved, then there might be some reason behind that conservation. So one reason was the functional tolerance, and another reason was that how it came into existence. Just because there are multiple different codons for the same amino acid, due to which there are chances of substitutions. Then compensatory mutations. So sometimes what happen is that in one place there is some substitution or mutation which might have drastic effect on the protein function or structure, but at the same time there is some other mutation which is occurring at some other position and which is compensating for this particular mutation. So for example, at position third, if proline is substituted with the glutamic acid, then similarly at sixth position, the glutamic acid is now substituted with proline. So in this case, there is a balance. So this is also possible that there are some compensatory mutations which are just uh, minimizing, or you can say, which is compensating the effect of particular substitution and restoring the protein function. Then fourth factor is functional advantage. So sometimes the substitutions may lead to the advantageous changes in the protein function, and due to which it promotes its conservation. So if particular substitution is advantageous, then it will be passed on to, means it will go across the evolution, it will remain conserved. So this is also one of the reason. Then neutral evolution. So neutral or nearly neutral substitutions, they can become fixed in a population due to genetic drift. So this might be the reason. Then evolutionary history. So substitutions occurring early in gene evolution may be conserved across lineages. So there are certain mutations which are occurring in the primitive ancestors, and now throughout the evolution, throughout the lineages, it is now conserved. It is not a new substitution which is being observed. It is retained from the primitive ancestors. So these are few of the factors which govern that particular mutation will be conservative or non-conservative, and what are the reasons that why that mutation is being observed, why it came into existence. So this was the patent which was filed, and from here I have obtained the table. So this was the table which I have shown in the previous session, and this particular type of substitution was mentioned as conservative substitution, that is from proline to glutamic acid. So in this, they have mentioned few things, out of which you can go through this if you want. But I want to say that how they have obtained that these mutations are conservative or these substitutions are conservative. So they said that the summary of the conservative amino acid substitutions were based on the blossom similarity matrix. So there are certain matrices in which for each substitution there is a score that is being assigned. So this blossom matrix, let's look into this. So this BLOSSOM stands for block substitution matrices, and these matrices were discovered by Dr. Stephen Henikoff and Dr. Georgia G. Henikoff. So it is also known as Henikoff and Henikoff, who have discovered this matrix. And these are the substitution matrices which are used in bioinformatics to score the amino acid substitutions in protein sequences. So now, when you see any multiple sequence alignment or even pairwise sequence alignment, so what is the main aim? Why do we go for sequence alignment? What do we get from there? So whenever two sequences are aligned, then there is some scores that are associated, because how can you judge that which alignment is better, which sequences are more related and which are less closely related? So all those things depends upon the scoring, that which kind of substitutions are favorable. So these all scores are provided by the BLOSSOM matrices. So there are generally two types of matrices at the level of amino acids. So one is the BLOSSOM matrices, and another one is the PAM, which is percent accepted mutations. So mainly the BLOSSOM matrices are generally used when you have to do such kind of comparisons. So in this, the scoring is done in this form, that the higher values indicate a higher likelihood of amino acids being conserved. So two substitutions, for example, alanine to some other amino acid substitution will be, if it is conserved, then it will have the higher value. And if or having a positive substitution, while if there is a lower value, which means the opposite. For example, some hydrophobic amino acid get substituted with some positively charged amino acid. For example, alanine gets substituted with some lysine. So now this will have the negative value because they both have entirely different biochemical properties, and there are many studies through which they have governed. There are various factors which through which it is being judged that particular substitution will be conservative or non-conservative. So in that way, by looking at that, what is the level of conservation, the scores are given. So if score is high, then it means that the substitution is conservative. And for this, how they have given the scores, because they have taken multiple protein sequences, they have performed multiple alignments, and at last they have performed their mean, they have calculated the mean, and then they have assigned the scores. So don't think that they have given the scores by looking at just one two sequences and then they have given the scores. No, they have performed the sequence alignment for the ensemble of sequences, and at last from they have concluded their results, they whatever things they observed, that so means it's not just on the smaller sample, but it was performed on the larger scale, and then these scores were given. That's why they have confidence. Now, if we say matrix names, so the BLOSSOM, if you see BLOSSOM anywhere, in even in the BLAST also, they also use BLOSSOM matrices. So in that, there are numbers which are assigned after this BLOSSOM. So what does it mean? So there are some integers, for example, BLOSSOM 62, BLOSSOM 45. These are different types of matrices which generally represents the scoring values. So it represents that which sequences were considered for identifying the scores. So 62 means the sequences which were taken, they were having the percent similarity of 62%. So it is the meaning of this integer. Similarly, matrix size. So it is a square matrix in which size indicates the level of sequence divergence. For example, BLOSSOM 62 for the sequences with the 62% identity. So in this, you see that which matrix you want to select. So for example, if you have sequences which share greater percent identity, so you will select greater BLOSSOM matrix. And when you have diverged sequences, for example, you have a sequence which shows less than 62% identity. So now you will not select BLOSSOM 62, but you will select some lower matrix such as BLOSSOM 45. So this is the reason behind these matrices. Then comes applications. So they are used in the sequence alignment algorithms such as BLAST to assess the sequence similarities and identify the homologous proteins. So homologous term we have also previously discussed. It means that the proteins that are coming from the same ancestor. So what happens is during evolution, there are certain neutral substitution, neutral mutations, and there are certain mutations due to which the proteins get diverged from one another. So in that case, to establish that either they are homologous or not, we can perform the sequence alignment. And after using these matrices, scores will be given to the sequence alignment, and you'll be able to know that okay, if the score is greater than this much percent identity, it means they share some regions in common, which means they are homologous proteins. So there is a criteria through which we decide that whether two proteins are homologous or not. Then what is the impact of this blossom matrix? So it has significantly improved the protein sequence analysis and evolutionary studies in bioinformatics. So in this I have provided you that how does this matrix appear. So in this there are rows and columns, and if you see carefully, then these rows and columns represents the single amino acid entities, and here is the score which is assigned for each substitution. So if alanine is substituted with alanine, then it will be given score as 4. Similarly, if alanine is substituted with arginine, so then it will be given the score as -1. So in this way, this matrix was being drawn. Here you can see that the frequently seen substitutions get positive scores. So the substitutions which are frequently seen, they will have positive score, which means that if they are frequently seen, that means there is some kind of benefit from those substitutions. That's why they are present, still present. So in that way, they are being given positive values. Then there are other substitutions which are rarely seen, so they will get negative scores because now the selection is acting negatively on them. Selection pressure is also acting on the sequences. So whenever some mutation or some change is not favorable for the individual, so what will happen? Selection will remove that thing. Those organisms will disappear and only the fittest will survive. So in this way, the substitutions which are rarely seen, they will get negative scores. Then substitutions which are expected by chance, they will get the score of 0. For example, here you can see, so this is this valine and threonine substitution. So this is observed by chance. That's why it has got the score of 0. Then self-match scores. So now in the diagonal, you'll see all the values given in diagonal, they are the self-match scores. And now these scores are also varying. So you might be confused that why these scores are varying so much. So the reason is that common amino acids such as alanine, isoleucine, leucine, valine, they have lower scores because they are very common amino acids. Whereas the rare amino acids such as tryptophan and those with the special roles such as prolines, they have higher scores. So this was the reason behind the scoring of this blossom matrix. Now let's quickly see one example that how this matrix is being used when we want to score some alignment. So for example, you have these two sequences and you have aligned them. So how you get the score at the end of alignment? So this is a 4 amino acid long protein. So this was just taken for your understanding. Otherwise, the proteins are very long. So in this case, firstly, we'll give score to the end-to-end substitution. So whenever n is substituted with the n, it has the value of 6 in the Blossom 62 matrix. So here we'll keep 6. Then if D is substituted with T, then what score we'll assign? So in this you see D is substituted with T, it will have -1. So we'll add -1 to this, then E with W, which means we have to see E, and then here W, it's -3. We'll add -3 to this, then Q with Q. So Q with Q will be having score of 5. So we'll add this, and at the end we'll get the score of 7. So in this way, we can perform sequence alignment and we'll get the score from the blossom matrix, and then we can compare those scores and we can get the knowledge or understanding that which two sequences are more closely related than the other ones. So in this way, it helps you to give the score to the alignment. Now let's move to the practice MCQs. So for today, the first question is, phylogenetic trees are often constructed by comparing the genome sequences of diverse species to find their relatedness. Based on your understanding, state whether the following statements are true or false. So here the two statements are given and you have to give this answer in terms of phylogenetic trees. You have to tell that either these statements are true or false. So statement 1 says that a larger number of differences corresponds to less relatedness among the species. Statement 2 says a smaller number of differences correspond to more relatedness among species. Now you have to think. So the answers are as follows. So the option A is both statement 1 and 2 are true. Answer option 2 is both statement 1 and 2 are false. Option 3 is statement 1 is true but statement 2 is false. And answer 4 is statement 1 is false but statement 2 is true. So now you have to tell that which one of the following will be the correct option given in the answers. So if you have the idea about the phylogenetic trees, then you might be aware that how these trees are drawn. Means we perform the sequence alignment, then from there only now we draw these phylogenetic trees. So read these statements and what do you think? So first statement is saying that larger number of differences correspond to less relatedness among the species. That means if more will be the differences, then the species will be less related to one another. That means they will placed far away in the phylogenetic tree. And if there will be small number of differences, then they will be more related. So it means that if you go for the sequence alignment, then the proteins which will be showing the greater sequence identity, they will be placed together. Whereas if they will be showing more differences, then they will be placed far away from one another. So now let's see what will be the right answer for this question. So the right option was option 1, which says that both these statements are true. And the reason is self-explanatory. Now let's see what are the phylogenetic trees and what points you should keep in mind when you go for and means go to see the phylogenetic trees. So first of all, phylogenetic trees, they represent the hypothesis about the evolutionary relationships among a group of organisms. So just focus on this word that they represent the hypothesis. What do we mean by this? So we means that as the new data comes and more analysis or more and more interpretations you perform, these phylogenetic trees are subject to change. So they can change. They are not fixed. They are not facts. So keep this thing in mind. Now the next point is the phylogenetic tree may be built using morphological, biochemical, behavioral or molecular features of species or other groups. So now the phylogenetic trees which we are discussing here, we are basically focusing on the molecular features of the species in which we are talking about the sequence level similarity and then drawing the phylogenetic tree. But there are other things also with the help of which we can draw these phylogenetic trees. So such as morphological or body shape. So by comparing the body shape among different organisms, we can also place them in the phylogenetic tree. Similarly, we can also look into the biochemical processes or biochemical features of the species, or we can also see their behavior. So in building a tree, we organize the species into the nested groups based on the shared derived traits. Derived trait means the traits which are different from those of the group's ancestor. So if there is a primitive ancestor and now the upcoming successor has gained some new feature or new trait, then we'll place it into the separate branch. We'll make two branch from the ancestor. One will be with the derived traits and another one with the some other trait if it exists. Then the sequences of genes or proteins can be compared among species and used to build the phylogenetic trees. So these things also we can do that instead of comparing the morphological characteristics, we can also compare their gene sequences and protein sequences, and then we can build the phylogenetic tree. And then the closely related species, they typically have few sequence differences, while the less related species tend to have more sequence differences. So this is the same point as it was mentioned in the statements in the question 1. Now I am giving you the brief picture of how the phylogenetic trees are constructed with the help of sequence alignment. So in this, for example, we have these four sequences. So they are the at the level of DNA nucleotide. These are the nucleotide sequences, and at the level of DNA. So for example, you have a gene, and for your understanding, I have given the sequence of length 10 nucleotides. So now how to calculate the how to draw the phylogenetic tree. So the first thing which you will do is you will look for the number of substitutions that are present in them. So for the multiple sequence alignment, firstly we'll make the pairs and we'll perform pairwise sequence alignment, and then we'll proceed with the matrix. So what you will do is that from each sequence, you'll make it pair with the other sequence, and then you'll look for the number of substitutions. For example, the pairs which will exist will be 1 2, 1 3, 1 4, 2 3, 2 4, and 3 4. So there will be six pairs which are possible from these four sequences. Now for each pair we'll look for the substitution. So for example, if you look at 1 and 2, then you will observe that you will look for the substitutions. So when they will be aligned, so firstly, if you see this third position, the C is being substituted with A in sequence number 2. So this is one substitution. Then if you move forward, then this particular sixth position in which A is substituted with T. So adenine is substituted with thymine. So there are two substitutions, and rest nucleotide sequence is exactly the same. So in this way, we got number of substitution as 2. Now we'll look for the fraction of these substitutions. So for that, we'll divide this number with the with the length, which is 10 nucleotides. So we'll get the fraction as zero point 0.2. Similarly, we'll calculate this fraction for all those pairs. And now by looking at this, we'll make a distance matrix. So distance is nothing. Distance means the number of substitutions. So this particular fraction is now denoting the distance between the two sequences. Because if you see carefully, then you'll observe that if the number of substitutions will be greater, so this fraction number is also higher. If number of substitutions are less, so then this number is also less. So in this way, for the matrix, what you will do is you'll place the number of sequences in both the rows and columns, and then you'll place the fractions. So for example, sequence 1 and 2, when you have compared them, what was the fraction which you obtained was zero point 0.2. So the distance will become zero point 0.2 and you will place it in the matrix here. Similarly, if you look here 1 and 2, then it will also be zero point 0.2. So the matrix which you will obtain from here, it will be a symmetrical matrix, which means that across the diagonal, the values will be the same. So that's why they have only placed the values in the topmost of the matrix. So with 1 and 2 it will be zero point 0.2, with 1 and 3 it is zero point 0.3, with 1 and 4 it is zero point 0.1. So in this way, for each of the pair, you will place the value in the matrix. Now the next step will be to check for the least distance. So let's start from the first. So here you can see that the sequence 1 and sequence 4 has the lesser distance, that is zero point 0.1 as compared to the others. So which means that they will be placed closer, because we have read from the hypothesis that the sequences which are related, more closely related, they show lesser difference in their sequence. So it will be 1 and 4 which are having lesser distance. Then if we look at the sequence 2, so then sequence 2 and 3, they show lesser difference. So it means that sequence 1 and 4 will be placed together and sequence 2 and 3 will be placed together. So in this way, you can infer a phylogenetic tree by looking at the distance matrix, and these distances are calculated on the basis of number of substitutions. So in this way, you can infer the tree. So there are multiple methods, out of which I have discussed only two methods. One is distance based and another one is the UPGMA, which is the unweighted pair group mean. So in this, what you do is same as this, you make the distance matrix. So greater number means larger difference and lesser number means lesser difference between the two sequences. So here also there are four sequences in the rows and columns, and these are the scores which are given. So all these matrices are the symmetrical matrices. That's why the scores are only placed in the one side of the matrix. And but what does it mean that in the top also the scores will be exactly the same as they are symmetrical matrices. So now how you will proceed with this? You will observe the matrix and you'll see that which of the pair have the least value. So in this, the least distance is between B and D, that is just 1. So as in this, we will pair or we will make the clusters. So now B and D will be clustered together. They are very closest. So they are clustered and then distance matrix is again recalculated. So this method is based on the recalculation of the distance matrix and clustering the individual sequences or the things which you have taken. So here we'll in the new matrix, now we will cluster BD together and now we'll look for the difference of these BD from other sequences. So how do we calculate the score? So for that, firstly you go to calculate the individual differences. So for example, if I want to know the difference between BD and A, so firstly I look for the difference between B and A and then D and A, and then I sum up both the differences and divide it with the 2. So here you can see that distance between A and B plus distance between A and D divided by 2, and this 2, why we use 2, because we have only two sequences. So it depends on the number of sequences which for which we are calculating this distance. So in this case, distance between A and BD will be 5. Similarly, distance between BD and C will be 3. So in this way, now you'll update the values or distance in this matrix. Now you will again you will again repeat the clustering process. Now you'll again see that which of the sequences have the least distance between them. So it's BD and C. So you'll cluster BDC now and place it here. And now you'll compare the distance between BDC and A. So how you'll check that? So you will calculate individual distance of A from B, D, and C. You'll add them and divide it with the 3, because now there are 3 sequences which are being paired. So you'll get the distance of 5. So in this I have written that how this 5 was obtained. Now there are no more sequences left for the clustering because we have got the idea. So now what we know is that firstly B and D are more closely related, then they are related to C, and then ultimately A is present. So in this way this tree can be drawn, and now these numbers represent the distance between them. So if you see here, the distance between C and A is 5. So now if you add these scores, so you will obtain the distance 5 from A to C. But how? So if you see this particular branch, it is 2.5. Now if we see this branch, this is 1. And if we see this smaller branch, it is 1.50. So if you add these numbers, it will add up to 5, which means distance between A and C will be 5. So in this way, phylogenetic trees are drawn with the help of distance matrices. And these distances are only known if you perform the sequence level alignments. So in this way, phylogenetic trees are drawn. Now let's move to question number 2, which says, among the options given below, identify the group of databases that cannot be used to analyze the human genome sequences. So now if you know about these databases, then you will be aware that um, which of them can be used to analyze the human genome. So let's see. So the first option is Ensemble Encode UCSC Genome Browser. Option 2 is Encode GenAtlas Ensemble. Option 3 is GeneCard RefSec GenAtlas. And option 4 is Encode GenAtlas and Flybase. So you can also use your common sense here. For example, if you don't have idea, then. So what do you think which would be the right option, which will not help you to analyze the human genome? So there are multiple options being given. So in this case, in these type of questions, you cannot see the first option and first part of the option and say that yes, if it deals with, then this option will be true. No, you have to go through each individual option. Then you have to make the inference that whether this option is true or not. So now let's quickly see that what will be the right answer. So the right option is the option number 4, which is encode genetless flybase. So why this option is correct? Because it has the database named as flybase, which is specifically made for the flies. So we cannot use it for analyzing human genome. Now let's see what the flybase provides you. It provides you the database of Drosophila genes and genomes. So these things you can do in flybase. It is not associated with the human genome. Now here I have provided you the list of other databases which were mentioned in that question. For example, if you don't have idea about any, so you can go through this table. So firstly, Ensemble, so it deals with multiple species including mammals, plants, insects, and many more. Then Encode also focuses on human and mouse with some data available for other species. Similarly, UCSC Genome Browser is also dealing with multiple species and they are emphasizing on the vertebrates, which involves human as well. Then Genetlas, it is also working on multiple species, but their focus is on human and some model organisms. GeneCard is also working on human with limited information on some other species. Then comes RefSec, which is having the wide coverage across species, including bacteria, archaea, fungi, plant, and animals. It also includes humans. And now, when we talk about flybase, then it is specifically for Drosophila melanogaster. It does not include any information related to any other species. So that's why the flybase was the right option. So I hope now you have got clarity related to these databases. You can also read the descriptions which I have provided. Now let's move to question number 3. So it says, which one of the following options is true with regard to the property of transposable elements in the genome? So if you know what are transposable elements, so then you will be able to answer this very easily. So option 1 is their movement within the genome is regulated by the host. Option 2 is they are absent in prokaryotic genome. Option 3 is the amount of transposable elements positively correlates with the genome size of the host species. And option 4 is all of these. So now you have to tell that which one of the following option is true when we say that this particular option is the property of transposable elements. Now let's see what will be the correct answer. So the correct answer is option number 3, which is the amount of transposable elements positively correlates with the genome size of the host species, which means that if there will be more number of transposable elements, then there will be the genome size will be larger. So that's why it indicates the positive correlation. Now I'll give you the brief picture of the transposable elements. So when we look at the human genome, this is specifically for the human genome. So there are only 1.5% of the genes which are protein coding in the human genome. Now rest 25.9% are the introns. And then these lines, signs, LTR retrotransposons, DNA transposons, these represents the transposable elements, and this codes for 45% of the human genome. So 45% of the human genome is attributed to the transposable elements. Then there are some other sequences such as simple sequence repeats, segmental duplications, miscellaneous heterochromatin, and miscellaneous unique sequences. These are also present. So 45% of the human genome is the transposable elements. Now, what are transposable elements? So they are also known as jumping genes, and these are generally the DNA sequences which moves from one location to another. So these were discovered in the corn, that is maize, during the 1940s and 50s by American scientist Barbara McClintock. So here I have provided her picture. So her work won her the Nobel Prize for Physiology or Medicine in 1983. So earlier she has discovered them in 1940s and 50s, but the people at that time, they were not accepting this work. They were a bit curious. They were not believing that this thing could exist. But with experiments and with lot more research, she could prove it, and then she have got Nobel Prize in 1983. Since McClintock's discovery, there are three basic types of transposons which have been identified till now. So which are class 2 transposons, then miniature inverted repeat transposable elements, also known as mites, or class 3 transposons, and retrotransposons. And among these three, class 1 and class 2 transposons are mainly observed and well studied. So class 1 transposons are the retrotransposons, and class 2 transposons are the DNA transposons. So keep this thing in mind that whenever a retro word is used, it means it has to do something with the RNA. So these retrotransposons, these are the transposons which firstly transcribes into the RNA intermediate, and then it undergoes reverse transcription and convert into the DNA intermediate, and then it will get integrated into some other target gene. So in this way, this is the mechanism that how these genes jump from the donor DNA to the target DNA. So they do not uh, individually go themselves, but they transcribes, and then that particular RNA intermediate is converting to DNA, and then it is getting integrated. Whereas the other type are the DNA transposons. So they are excised from the donor DNA, and then that particular intermediate is now integrated into the target DNA. And in this way, this particular mechanism, if you see carefully, it is the cut and paste mechanism, where the transposon is being cut and then pasted. But this mechanism, if you see, it is like copy and paste, means it is still present here and it is copied and then it is pasted. So in this way, donor DNA and target DNA both contain the transposable element, whereas in DNA transposons, only the target DNA is containing the transposable element, and donor is entirely giving its transposable element to the target. Now, just for the more clarity, you can see this. So class 1, as I have mentioned, that they are the retrotransposons. So they are categorized as LTRs, then line, sign, they come under retrotransposons. Whereas in the DNA transposons, the types are TIR, Helitron, and Maverick. Now these transposons, they have the LTRs, long terminal repeats, and in between them, there could be some coding gene that could be present. So these GAG, POL, it represent the genes which can synthesize some proteins. Similarly, in lines, there are proteins such as GAG and POL gene which are present, and then poly A. In sign also, it has left monomer A rich, right monomer, and poly A. So not all the retrotransposons can code for some protein, but there are few which can code. Whereas in DNA transposons, the genes which are present is transposases, replicase, helicase, integrase, polymerase. So these names appear as if they are some enzymes. So similarly, these genes code for these enzymes, hence they have got the name as this: replicase, helicase, integrase, polymerase, and transposase. So as I've discussed earlier, that these retrotransposons, they are based on copy and paste mechanism because they are converting to RNA intermediate, and then after reverse transcription they are converting to DNA intermediate. Similarly, in DNA transposons there is excision and then integration. So here I've mentioned that what does these mean, what are these genes, and what are their functions. So you can go through this. Now, why these transposable elements are important? Because in the question we have seen that they are positively correlated with the genome complexity or genome size. So now what they help in? So firstly, they are advantageous for the genome plasticity, which means they promote the genome size expansion. They cause alternative splicing and exonization. So these transposable element domestication, it alters the gene expression, it restructures the regulatory network, it influences epigenetic control. So all those things are brought about by the transposable elements because these are the jumping genes. They can move from one place to another. Then host range. So it influences the host range of strains through its overall effect on pathogenicity. Varied repeat elements observed in the strains of different host ranges. Then they are also important for the evolution because they affect host fitness through deleterious or advantageous insertions. Two-speed genome concepts highlights fast evolving LS genome compartment that is rich with the transposable elements, and they drive speciation and adaptive evolution. So they are also responsible for the evolution. Then for pathogenicity, so proximity with the avirulent pathogenicity associated genes confers gained virulence through deletion of avirulent genes, promotes pathogenicity by gained nucleotide diversity. So in this way, they are helping in these four things. Similarly, I've given here the same picture which I've shown earlier, the distribution of these line, sign, and the other DNA transposons in the human genome. So I hope now the concept of transposon is cleared. So let's move to question number fourth, which says which one of the following refers to the speciation due to geographical isolation. So I hope you are aware of this term speciation. So this speciation means that from the ancestor, when two different species arise, so that is known as speciation. So means arising of new species is known as speciation event. When new species are coming, so here you have to tell that which one of the following speciation event refers to as the speciation which happens due to geographical isolation. So option 1 was sympatric speciation, option 2 is parapatric speciation, option 3 is peripatric speciation, and option 4 is allopatric speciation. So what do you think, which kind of speciation happens due to the geographical isolation? Okay, now let's see what is the correct option. So option 4 is the correct, which is allopatric speciation. So the speciation which occurs due to geographical isolation is allopatric. Now I'll give you the idea regarding all other substitutions, all other speciation events and how they arises. So firstly, allopatric speciation, it arises because of the geographically isolated or geographically the barriers which appears due to which the two species they now the gene flow is prevented because they are separated with the geographical barrier, and it could be the mountains or there could be some river that is flowing between them due to which they are not able to cross the area and cross this barrier, due to which the gene flow is hindered. They cannot flow their gene among one another. With time, they will accommodate some genetic differences and ultimately they will become entirely distinct species and they'll not be able to interbreed. So when we say species, when we say the particular organisms belong to a species, when they can interbreed and produce a viable offspring. So if they cannot produce viable offspring or if they cannot interbreed, so we'll call them as the distinct species. Then if we look at the sympatric speciation, so this is the speciation event in which the new species arise within the same geographical area. So here the geographical area is separated by the barrier, but in sympatric the geographical area is the same as the parent population, but it often involves the development of reproductive barriers. So the barriers are at the level of reproductive barriers which limit their interbreeding between the subpopulations within the same region. So now the differences they arise at the level of reproductive barriers, due to which they become two different species. Now let's see the parapatric speciation. So parapatric speciation occurs when the population are adjacent to each other but have limited gene flow. In this also they are adjacent to one another, but the gene flow is limited due to specific factors such as environmental gradient or barrier. So there is the environmental gradient or barrier is present, due to which over generation divergence may result in distinct species. For example, they are living in the same area, so there is a water body present and there is a land that is present in the same geographical area. Some species move to the land and some species they are remaining in the water body, due to which although they are present in the same geographical area, but still there will be differences among them, due to which they will diverge to the distinct species. Now there comes the peripatric speciation. So it is the speciation event in which the small population becomes isolated from the main population, typically due to dispersal to a distant location or habitat shift. So in this, they are becoming isolated from the main population because they are now moving to some distant location. They are shifting their habitat. So now they'll not be able to uh, maintain the gene flow among one another. So the isolated group can undergo the unique evolutionary changes and potentially leading to speciation. So this peripatric and the allopatric speciation, they are nearly same, because in this also there is geographical barrier, and in this also as they are shifting, so there is the geographical barrier. But the main difference between them is that in peripatric the speciation is coming at the periphery. That means only a small amount of population is moving out, but here the population is, means greater size of population, almost equal, is being divided with the geographical barrier. Similarly, parapatric and sympatric also, the parapatric only represents the smaller fraction, whereas in sympatric, this fraction is greater. So they both are present in the same geographical region, whereas they both are present in different geographical areas due to the barriers which are created between them. So this is all about the speciation categories and their definitions. So I hope now you have got the clarity regarding the different types of speciation events. So let's move to next question. So the question 5 is, which one of the following statements is incorrect with regard to phylogenetic trees? So option 1 is, they represent the facts since supported by the functional genomic data. Option 2 is, they represent the hypothesis and supported by the functional genomic data. Option 3 is, the branching in a phylogenetic tree reflects how species evolved from a series of common ancestors. And option 4 is, rotating a tree about its branch points does not change the information it carries. So you have to tell which option is incorrect. So by now we have discussed a lot about phylogenetic trees. So I hope you will be aware of the correct answer. So let's see. Option 1 is the correct option. That is, they represent facts since supported by the functional genomic data. Because as I have previously discussed, that the phylogenetic trees, these are not the facts, but these are the hypotheses. So that's why this option will be true, as it is incorrect. So in this, if you see the fourth option, which is saying rotating a tree about its branch point does not change the information. So I'm giving you the idea that how it works. For example, if you rotate this tree about this particular branch, so it will appear like this now. This Z will shift to this position, but still the information which it is giving will be same, which means that Z and Y are more closely related, then these two are closely related to X, and then all of them, these are related to W. So in this way, it still gives the same information. It is the same tree. So here, different examples of rotations are being given. So in different ways, it could be rotated about the branch. It can be rotated from here or here or here. So in this way, all of them will be the same trees. Now let's move to the next question. The question 6 says, the field of metagenomics involves. So we have also gone through the term metagenomics. So now let's see the options. Option 1 is the study of genetic material recovered directly from the environmental samples. Option 2 is computational analysis of the human genome. Option 3 is sequencing the genome of the gut microbiota. And option 4 is sequence analysis of the transposable elements of the human genome. So now you have to tell that what does metagenomics involve. So when you go through these options, you will get the idea. Now let's see which option is the correct. So the correct option is the study of genetic material recovered directly from the environmental samples. So that is referred to as metagenomics. But there are other options which might be confusing, such as this option number 3. So we'll discuss this in detail, that why this option is not true. Now let's see. So the metagenomics, it refers to the study of genetic material recovered directly from the environmental samples. So environmental samples can be soil, water, or human gut, and it allows the scientists to explore the genetic diversity and functional potential of these microbial communities without the need of isolating and culturing individual organisms. So instead of taking the information from individual organisms, we are taking the information from the environmental sample in which there will be multiple individuals that are present. So why other options were not accurate? So option 2 was computational analysis of human genome. So when we say metagenome, it generally refers to the multiple genomes that are being studied and not just the human genome. That's why this option was not correct. Then sequencing the genome of the gut microbiota. So yes, this gut microbiota also comes under metagenomics, because here we have discussed that different environmental samples are taken and one is the human gut. But why it is not accurate? Because this metagenomics is not limited to the gut microbiota. There are other samples which are taken from soil or water. So that's why this option was not accurate, but option 1 was accurate, which means that we are taking the information of the environmental samples. Then the sequence analysis of the transposable elements of the human genome. So in this also, this particular study is focusing on the transposable elements of human genome. So it is just considering the human genome. So in this way, it cannot be the part of metagenomic analysis. Now, these are few applications of these metagenomics. So, metagenomics can help in agriculture, biotechnology, textile industry, food sector, functional gene and metabolic pathways, explore microbial diversity, disease diagnosis, human and animal genetics, and medicine and antibiotics. So, in these fields, metagenomics is generally used, it is applied, and it is helping in these fields. Now, let's move to next question, which says, a multiple sequence alignment analysis helps us to understand. So, we have discussed it in the previous session as well, that why we perform multiple sequence alignments. Option A is ancestral relationships. Option 2 is regions under lesser selection pressure. Option 3 is regulatory elements in the genome. And option 4 is all of these. So, what can you understand from the multiple sequence alignment? Okay, so the correct answer is all of these. You can study all these things, you can understand all these things by using multiple sequence alignment, that is ancestral relationships, the regions which are under lesser selection pressures, and the regulatory elements in the genome. So here I have provided you few applications of this multiple sequence alignment. So first of all, the first application is the phylogenetic analysis, which means that you are aligning multiple sequences from different species, and then you can infer the evolutionary relationship. So it is based on the principle that if the sequences which you have taken, if they show lesser difference, then they will be closely related as compared to the sequences which show greater differences. Then comes protein structure prediction. So you can also predict the structure of the protein by performing multiple sequence alignment. So it is based on the principle that the sequences which show similarity among different species, it means those sequences are preserved, or you can say those sequences are conserved, and they might contribute to some motif or some functional domain which is critical for the folding or function. So in that way, you can predict the structure of that particular region. For example, you have the unknown sequence and you want to predict the structure. So you'll align it, you'll perform multiple sequence alignment, then you'll see that to which part of the sequence of the known species it is aligning, and with that, you can predict the structure of that unknown region. Then functional annotation, it is also done in the same way. You align multiple sequences, and for the sequence for which information is not known, you'll see that to which part of the known sequence it is aligning. So the functional information of the known sequence will be used to predict the unknown sequence's function. Then identification of conserved elements. So you can also identify the conserved elements, because the regions which are showing lesser differences or lesser number of substitutions, they will be highly conserved. and they might be coming from the promoter sequences, binding sites, or the functional motifs, because these are the regions which are mainly conserved because they have their application, or they have their effect on the protein functionality. Then drug discovery and design. So you can also use the multiple sequence alignment in identifying the conserved drug binding sites or the on the proteins, which will help you in the rational drug design and understanding the effects of genetic variations on the drug responses. Then you can also perform genome annotations. So annotations is nothing but it is the adding details. So the same thing you can do by aligning, because if you have the new genome sequence, so the new sequence of the gene, you'll align both the new and the known sequence, and then you'll see that where they are aligning with the greater identity or greater similarity. So those regions will be either the regulatory regions or some motifs or some domains. So in that way, you can annotate that particular region. You can predict the structure, regulatory elements, and you can also predict the function of the gene, which is unknown. Now let's move to next question. So which one of the following options is true statement for homologous recombination process? Option 1 is it typically occurs during mitosis. Option 2 is it involves the exchange of genetic material between the non-homologous chromosomes. Option 3 is it can result in genetic diversity. And option 4 is it only occurs in prokaryotic cells. So I hope you have the idea regarding the homologous recombination. So what do you think? Which could be the true statement? So let's see what is the right answer. So the right answer is it can result in the genetic diversity. So let's look into the other options that why they are not true. So they are saying it typically occurs during mitosis in option 1. So mostly homologous recombination is observed in the meiosis. We do not say that it is not observed in mitosis, but it is mainly observed during meiosis. So we cannot say that it typically occurs during mitosis because it is typically occurring during meiosis, not mitosis. So that's why this option was not true. Then the second option says that it involves the exchange of genetic material between the non-homologous chromosomes. But this particular homologous recombination process is very specific to the homologous chromosomes. So in this way, option 2 is also not correct. Then if we look at option number 4, so they are saying it occurs only in the prokaryotic cells. So this is also not true because it is also occurring in the eukaryotic cells. So in this way, the correct option will be option number 3. So homologous recombination is a genetic process which involves the exchange of genetic material between the homologous sequences, which means which are similar sequences often between the homologous chromosomes during meiosis. And this process can lead to genetic diversity among the offsprings due to the recombination or reshuffling of the genetic material. So here I have provided you the brief picture. So in this you can see that this is the paternal chromosome, this is the maternal chromosome, these are the homologous chromosomes. And then after the round of DNA replication, they both will be means they will be replicated. The DNA, these chromosomes will be replicated. There will be the two copies of each chromatid that is present. Now the crossing over will happen during the meiosis, and at last recombined chromosomes will be obtained. So in this way, you can say that the nucleotide sequences are exchanged between the two similar or identical molecules of DNA, and then this exchanging of DNA is important source of the genetic variation among the offsprings, due to which we look slightly mixture of our mother and father. We are not exactly to either parent, but we are the combinations. So there are certain differences which are observed in us. So that all these differences are the result of this recombination. So let's move to the next question. So here is the question number 9, which says the term comparative genomics refers to the field of study in which the genomic features of different organisms are compared. Which one of the following options may not be the direct outcome of comparative genomics? So in this, they have already provided you with the information of the comparative genomics, that what does it mean, and you have to choose the option which is not the direct outcome of this comparative genomics. So option 1 says identification of regulatory elements of the genome. Option 2 is understanding the evolutionary changes in the genome. Option 3 is phylogenetic position of species. And option 4 is epigenetic changes. So what do you think, which of the option is not the direct outcome of the comparative genomics? So let's see its answer. So the correct option is epigenetic changes. Rest all the three options, these are the direct outcomes of the comparative genomics, but epigenetic changes we cannot directly observe from the comparative genomics. Now let's see what does comparative genomic means. So in comparative genomics, you are generally comparing the multiple species genomes from the multiple species. So researchers choose the appropriate timescale of the evolutionary conservation for the question being addressed, what question they are asking. So they can look for the common features of different organisms. So in this, they have considered different organisms, entirely different, and they are looking for the common features which are present among them. So this is the common feature. For example, human and fish are often encoded within the DNA evolutionarily conserved between them. Then what else they can do? They can also look at the closely related species such as human and chimpanzees and show which genetic elements are unique to both. So earlier they were focusing on entirely different organisms. Now they are focusing on closely related organisms and they are identifying the regions which are leading to the uniqueness among all of them. So this can also be done with the comparative genomics. Then another thing is genetic differences within one species, such as our own can reveal variants with the role in disease. So in this, they are comparing the one species only, means just the human, and then they are looking for the variants and how those variants are causing some disease. For example, a patient and normal person, so there when their genome is being compared, so that will be referred to as comparative genomics, and it will give you the idea related to the variants which are causing some disease. Now let's see what does epigenetic means and why epigenetic changes cannot be observed directly from the comparative genomics. So the reason is that these epigenetics means uh, it is the study which is focused on the changing in DNA that do not involve alteration to the underlying sequence. So in comparative genomics, you are dealing with the genome sequence, but epigenetic changes they are not occurring on the sequences, they do not alter the sequence. So the genome will appear although it appears similar, but there will be different epigenetic changes that are acting on them. And this field is also known as epigenomics. So epigenetic is the study of how your behavior and environment can cause the changes that affect the way your genes work. So it is also possible that you and the other person they have the similar genes that are present in both of them, but in one that particular gene is not able to express because he is exposed to some different environment, whereas you are exposed to some different environment. So in your body, that gene is expressing normally. So this is known as epigenetic changes. And unlike the genetic changes, these epigenetic changes are reversible. They can reverse back. So they do not change the DNA sequence, but they can change how your body reads the DNA sequence. So certain epigenetic modifications may be passed from the parent cell to the daughter cell during cell division or one generation to the next. So yeah, they can be passed. They may be passed from parent to offspring. Now I've given you few examples that what is the epigenetic changes that are generally observed in the body. So nerve cell versus muscle cell. So muscle cells and nerve cells they both have the same DNA, but when you will observe the proteins and genes which are expressed among them, they will be entirely different. So nerve cell it transports the information to the other cells in your body, whereas muscle cell has the structure which aids in your body's ability to move. So epigenetics or the changes which are occurring due to these epigenetic thing which is occurring due to the environment, it allows the muscle cells to turn on genes to make proteins which are important for its job and turn off the genes which are important for the nerve cells job. So our cell is very smart, our body is very smart, it do not express the genes which are not required because this expression is very expensive. There are different genes which are required for this expression, so our cell don't waste anything. It is very much, you can say it's very efficient. It expresses only those genes which are required and it turn off the other genes. So these all things they are only possible with the help of epigenetics because the DNA is same in all the cells, but which gene are expressing in particular cell, it is governed by these epigenetic factors. Then there is another study which was performed for the newborn 26-year-old and 103-year-old people, and in that these epigenetic changes were being analyzed. So in this, the epigenetic change which they selected was DNA methylation. So they observed the DNA methylation at million of sites among all these three categories, and they observed that the level of DNA methylation, it decreases with age. So in that case, the newborn were having greater DNA methylation as compared to the 103-year-old person, whereas this 26-year-old, it was having the intermediate number of DNA methylations that were observed. So in that case, these epigenetic changes, the one of the factor which governs is the age as well. So now, what are the different epigenetic modifications that are possible? So one is DNA methylation, which can lead to gene silencing if there are hypermethylations. They can also lead to gene activation if there is hypomethylation. And if you see in the cancers and neurodegenerative diseases, so you will see that there is some disturbances or some differences in the DNA methylation pattern, which leads to these cancer and neurodegenerative diseases. Then comes histone modifications. So similarly, if acetylation of particular histones will be done, then it will activate some gene. Whereas if the methylation will be done, so it can activate or repress some of the genes. So it is also linked to cancer and cardiovascular diseases. So here you can get the idea that these epigenetic changes are very important. If these are disturbed, then they will lead to some disease. They can cause cancers, cardiovascular disease, neurodegenerative disease. Similarly, non-coding RNA regulation, so that is also regulated with the epigenetic modifications such as mRNA degradation and inhibition of translation. So, when you see the gene silencing with the microRNA and the small interfering RNA, so that is also some kind of epigenetic modification in which the particular mRNA is being degraded or either it is repressed so that it could not translate. So it is also associated with the cancer and neurodegenerative disorders. Then X chromosome inactivation. So its inactivation is also the epigenetic modification which leads to this inactivation, and it helps to ensure the dosage compensation. And if there will be aberrations, then it will lead to some disease. Then development and differentiation. So these development and differentiations to establish the cell identity is also with the help of epigenetic modifications, and they are very crucial for the normal development and tissue specific functions. So this is the these are the epigenetic modifications that can be observed, and this is their consequence and their impact on health and disease. Now, here I have given you the idea that what are the different epigenetic mechanisms, and they're affected by different factors. So these factors are the development. For example, the individual which is now inside the utero, it will have some different epigenetic mechanisms, whereas in childhood it has some different mechanisms, and with age it will have some different epigenetic mechanisms that are working on him. Then environmental chemicals. So if you'll be exposed to some chemicals, then there will be different epigenetic modifications which will be observed in your genome. Drugs and pharmaceuticals, they are also modifying your genome to some extent. They are also bringing about these epigenetic changes. Then aging is also the one of the factor. Then diet. So in this you can see that the diet, environment, and all these factors, they play major role in the epigenetic modifications. So in this, DNA methylation can also be observed, or histone methylation or acetylation can also be observed. So these factors, their roles I've already discussed. And then what are their health endpoints? So they can lead to cancer, autoimmune disease, mental disorders, diabetes. Now I want to address one of your question which you asked related to DNA methylation. So when I was saying DNA methylation, so firstly I'll tell you that what does DNA methylation means. So DNA methylation means there are some nitrogenous bases which can get methylated. So particularly when we say DNA methylation, so we are talking about the cytosins now which are methylated. And why this DNA methylation is affecting the functionality of a gene? So these CPG, so firstly I'll tell you the CPG islands. So CPG islands are nothing but these are the repeats of the cytosine and guanine which are generally observed at the promoter region of the gene. So promoter region is the region to which the transcription factors are binding or some other factors are binding which are ultimately leading to the expression of that gene. So now, if these particular cytosins or guanine molecules which are present, these cytosins, if they will be methylated, so now it will avoid binding to the transcription factors or the activators, and the gene will not be able to express properly, due to which the particular gene, it will be repressed, its expression will be hindered. So in that way, you'll see that this particular DNA methylation, it has its effect on the expression of the gene. And there was another question, so in which somebody has asked that when thiamine is converting to uracil in the RNA, so as we know that if we look carefully at the structure of thiamine, there is one methyl group which is present, whereas in uracil that methyl group is absent. That is the main difference between the thiamine and uracil. So somebody asked that is it the same demethylation pattern which is observed, removal of methyl group when the DNA is converting to RNA? So for that, I want to answer that that does not come under the epigenetic modification. That are the normal nitrogenous bases which are present. So when we say DNA methylation, so we are focusing on the methylation at the cytosine nitrogenous base. We are not talking about the thiamine or uracil conversions. So um, in that way, these DNA methylations, they have their effects on the regulation of the gene. They can either express gene or they can either repress the gene. So if there will be more methylations, then the gene will not be able to express properly. Its expression will be affected. Similarly, in histone modifications also, you can see that if there will be acetylation of the histones, so it will lead to the greater expression of those genes. And there are certain regions in the chromosome. When we say chromosome, so these chromosomes are the DNA molecules which are wrapped around these histone proteins very closely. So there are certain regions which are very compactly packed. Those are known as heterochromatin, whereas there are certain regions which are accessible, in which the DNA is accessible and genes are active. That is known as the euchromatin part. This one is the heterochromatin part. So if in the heterochromatin part the histones are very compactly arranged, so if there will be acetylation, then these histones will move slightly away from one another. It will relax this structure, it will convert to euchromatin, and now these genes will be able to express as this DNA will be accessible. So this is the impact of histone modifications on the expression of gene, and this is the one of the epigenetic modification only. Now let's move to next question. So which among the following option will not be much useful to study comparative genomics? So option 1 is whole genome analysis, option 2 is gene clustering by function, option 3 is antibody screening, and option 4 is gene sequence variation study. So what do you think, which of the option will not be useful when you are studying the comparative genomics? So we have already discussed about the comparative genomics, what are its applications and how we perform this. So now I hope you'll be able to answer this question. So let's see what is the right answer. So the right answer is option 3, which is antibody screening. Because for comparative genomics, you use the whole genome analysis, you also cluster the genes by function, you also sequence the gene variations, but you do not do antibody screening because it is entirely based on the chromosomes or the genomes. So we do not require antibody screening here. So now there are different ways to study comparative genomics. First one is sequence alignment and comparisons. So sequence comparisons can be at the level of multiple sequence alignment, where you are aligning multiple sequences from different species, or pairwise sequence comparisons, which are in which only two sequences are being compared, or BLAST. So for example, if you have some unknown sequence and you do not know to which it has to be compared, so you'll perform BLAST. It will search it to the entire database that is available. Then comes phylogenetic analysis. So after alignment, you can also construct the phylogenetic trees, and for that you can use software such as Phylip, RaxML, Mega. You can also perform the use the comparative genomic databases. You can assess the genomic data. You can compare the genomic annotations in the comparative genomics. Now, function annotations, you can also predict the gene functions. You can identify the conserved domains which are being conserved among different species. Then you can also study gene orthology and paralogy. So we have also discussed these terms. What does orthology means? What does paralogy means? And we'll discuss them in detail in the upcoming slides. So these things also you can determine with the help of the comparative genomics. Then gene expression analysis, you can also analyze the expression of genes in different species. You can compare the gene expression profiles. You can use the RNA-seq and expression data to make comparisons. Now let's look at the antibody screening. So in that we saw that there was antibody screening which we do not use for comparative genomics. So here I've provided you different methods for which this antibody screening is used. So first one is the enzyme linked immunosorbent assay, for which you use the antibody screening, and this method is involved in the detection and quantification of specific proteins, antigens or antibodies in the biological samples. So for that you require antibody screening. Then in Western blotting, which is also known as immunoblotting, in which you identify and characterize specific proteins based on their reactivity with the antibodies. Then immunohistochemistry. For that also, if you want to visualize and locate specific antigens or proteins within the tissue sections to study their distribution and localization, you will perform the antibody screening. For flow cytometry, identify and quantify the cell surface markers or the intracellular proteins in the single cells or the cell populations. You need the antibody screening. Then antibody microarrays. So for that also you'll use the antibody screening, which are the high throughput screening of antibody antigen interactions for the applications like biomarkers discovery or disease diagnostics. Then for fast display, that means identifying the antibodies or the peptides with specific binding affinity to a target antigen often used in the drug development and research. So for all these fields you use the antibody screening. Now let's move to the question number 11. So, what does the term phylogenetics refer to in the context of comparative genomic analysis? So, option 1 is the study of genetic mutations within a single species, the study of evolutionary relationship between species, the analysis of gene expression in different tissues, and the study of physical structure of genes. So by now, I hope it will be very clear to you what does phylogenetic means in context of comparative genomics, because we have discussed about it many times. So let's see the correct answer. So the correct answer is option number 2, which means the study of evolutionary relationships between the species. So which is referred to as phylogenetics. It is the study of evolutionary history and relationship among species, which is often based on the genetic data such as DNA samples. So now let's move to question number 12. In comparative genomic analysis, orthologous genes are the genes that option 1 is perform similar functions in different species due to the convergent evolution. Option 2 is have the same sequence in all the species being compared. Option 3 is are homologous and derived from a common ancestor. And option 4 is are only found in a single species. So if you are aware about the term orthologous, so you will be able to answer this. Read each option carefully and think that what would be the right option. Now let's see. So the correct option is option number 3, which says that orthologous genes are the genes that are homologous and derived from a common ancestor. So what option 1 says? Perform similar functions in different species due to convergent evolution. So when we say convergent evolution, it means that the particular gene is coming from two different ancestors. But orthologous, its main definition is that it has the common ancestor. So option 1 is incorrect. Then option 2 is saying have the same sequence in all the species being compared. So no, this is also not true. It is not true that always the same sequence which is being observed it means orthologous, because same sequence can also arise due to the convergent evolution. That means the ancestors are not same, but still the sequence similarity exists. So that can also be possible, which is the paralogous gene. So we cannot say that the same sequence in all the species is the orthologous genes. Similarly, are only found in single species. So that's also not true, because orthologous genes are the genes which are coming from the common ancestor. So they are involving multiple species. We cannot say orthologous by looking at single species. Now let's go to the different terms which are confusing so that you can get clarity. So when we say homologs, so it is a very broad term which refers to the genes, proteins or sequences with a shared and evolutionary origin. That means their origin is same. They can be proteins, they can be genes, they can be sequences. So in example, we have protein A and B with similar sequence and it is having the similar origin, so it will be a homolog. So when we say ortholog, it is a subcategory of homolog. That means its ancestor is same, but these genes are in different species have evolved from the common ancestral gene due to speciation events. That there was common ancestor. Now there occurs the speciation event due to which two different species are formed and they share some kind of sequence similarity. We cannot say that the sequence will be entirely similar, but still there is the similarity. The origin is same. Origin is the single ancestor. For example, human insulin gene and the mouse insulin gene is the good example of these orthologs. Then if we look at the paralogs, so the paralogs are the genes. It is also the subcategory of homologs. These are the genes within the same species that arose from the gene duplication events. So whenever a gene undergoes duplication, it becomes the paralog. So human hemoglobin genes, alpha and beta globins, these are the paralogs of one another. They occur due to the duplication. Then comes xenologs. So genes in the different species that share the common ancestry but have undergone horizontal gene transfer events. So in this also the ancestor was same, but the transfer of this gene has occurred due to the horizontal gene transfer. For example, bacterial antibiotic resistant genes. So they are also the example of xenologs. So for example, the E. coli, it has some antibiotic resistance that is present in it. And now if it passes to the another bacteria, that particular resistance, so that transfer will be the horizontal gene transfer, and the that particular resistant gene, it will be known as the xenolog. So those two genes will be referred to as the xenolog of one another. Then analogs. So analogs means the genes, proteins, or sequences which perform the similar functions but have distinct evolutionary origins. Means their origins, their ancestors are entirely different, but they perform similar functions. For example, wings of bats and wings of birds. So they are their ancestors are different. One are mammals, other are reptiles. But if you look at their wings are performing similar function. That means which are helping them for flying. So it appears that they have the common origin, but it is not true. They both have the different origins, but still they appear as the similar functional gene or similar functions. So they are converging to some function. So this is analogs. Now here I've provided you the example. So there was some ancestral histone gene, that was H1 gene, which was present. It has undergone duplication to H11 and H12. And then there occurred the speciation event in which human have diverged from the chimpanzee. Both contains the same ancestral gene histone, now which might have similar sequence, and it has accumulated some of the genetic differences as well. So these two genes will be referred to as paralogs, because in within single species, when some gene has undergone duplication, it is known as paralog. When those genes are coming from the same ancestor in two different species, they are called the orthologs. And when this particular gene is sharing its sequence or its function to some other protein which is coming from some other ancestral gene, for example, the ancestral gene is HNS gene, that is forming the bacterial HNS protein, which is very similar to this human histone. So these two proteins will be analogs because they have different origins. They are not sharing their origin, but yet they are performing similar function. So they appear as if they have converged their function. So these will be the analogs. Now let's move to the next question, which says, what is the main advantage of using the model organisms in comparative genomic analysis? So why do we use the model organisms? So option 1 was saying they have larger genomes, making analysis easier. Option 2 is they are more closely related to humans. Option 3 is they have simpler biological systems for easier study. And option 4 was they are more abundant in nature. So, what do you think? Why do we use model organisms? Why do not we go on the analysis on the complete individual? Why do we go for the model organisms? So, what do you think? Okay, now let's see what will be the correct answer for this question. So, the correct option is option number 3, which says they have simpler biological systems for easier study. That is the main purpose why we select model organisms, because if they have larger genome and analysis, so first of all, if they have larger genome, then analysis won't be easy, it will be difficult. Then they are more closely related to humans. Yeah, this is also true. They are more abundant in nature. This is somewhat true, but the most accurate will be they have simpler biological systems for easier study. So here I have provided the list of different model organisms which are being used. This one is in the plants, so that is Arabidopsis thaliana. This is in the animals, both vertebrates and invertebrates. So in vertebrates, the species which come are the Mus musculus, Danio radio, and X tropicalis. Whereas in invertebrates, the species which the model organisms which are present are Drosophila melanogaster, also known as fruit fly, C. elegans, E. coli, and Saccharomyces cerevisiae, which is the yeast. So in this, if you see the topmost Orange colored box represents the genome size of that particular organism, and the bottom represents the protein coding shared with the humans. So how much genome percentage is shared, protein coding part is shared with human versus matching genes which are associated with the human disease. So out of them, how many genes were associated with the disease? So if we compare all of them, we'll see that the majorly the mouse shares the protein coding with the human at the greater percentage, that is 90. Then this frog and then Drosophila. Then there are 65 of the mouse genes which are common between human and mouse which are associated with the disease. Then 84 in the Danio radio. That means there are 84 genes which are present in both human and this zebra fish and which are leading to the human disease. So if you see here, it's written 70%. That means this is the protein coding that is being shared. And here it is telling you the genes which are associated with the disease. So might be those extra 14 genes, they do not share the protein coding similarity with the human, but still they are involved in the human disease. Then similarly for the frog, the fraction is same. And this way, these are the different model organisms that are being used as they share greater similarity to human and they have their genome size is lesser as compared to human. So due to which we can easily interpret the things. So there are different model organisms such as yeast. What are the advantages? So it is the single-celled eukaryote which is grown in vitro. It replicates quickly and genes are easy to manipulate. That's why we select this. Then why we select zebrafish? So its embryos and larvae are transparent and we can use the fluorescence as a visual tool, and it is also useful for the transgenic models, due to which the zebrafish is selected as the model organism. Then comes mouse or mus musculus. So it is genetically similar to humans as we have seen here also. Inbred colonies have limited diversity and new strains are immunodeficient. So, due to which mouse is also a better model organism. Then macaque monkey, genetically similar to humans, similar immune response to humans, and useful for the longitudinal studies. So, for this, we can also select this monkey as the model organism. Now, let's move to next question. So, which says which technique is commonly used to perform comparative genomic analysis? So we have gone through this. Option 1 is polymerase chain reaction, option 2 is DNA sequencing, option 3 is Western blotting, and option 4 is ELISA. So what do you think? Which of the technique is commonly used? Now I want to tell you that whenever you read the question, don't do hurry and tick the option, but read it carefully because each single word can change the entire meaning of the question. So in this, they are saying commonly used. And there are certain questions in which, when you read them, you will see that there will be more options, which will be confusing, which will appear as if both are the true. So, how you will select one among them? So, now let's see the correct answer. The correct answer is DNA sequencing. Many of you will be confused that why not polymerase chain reaction? Because that is the first step which we perform whenever we perform genome analysis. Why is it DNA sequencing? So now to answer this, when we are performing comparative genomic analysis, that means we'll be comparing the DNA sequences from multiple species. So the DNA sequencing is the commonly used. Polymerase chain reaction, this is generally done to amplify the sample which you have, but it is not very commonly used. The most common among all of these is the DNA sequencing, which is most common. Now we have discussed all those techniques except ELISA. So here I've given you the information about ELISA, which stands for enzyme linked immunosorbent assay, and we have discussed about it a very little when we were talking about antibody screening. So why do we use ELISA? So it is generally used to detect and quantify the specific proteins, antigens or antibodies which are present in the biological samples. Now, what are the components of ELISA? So you need microtiter plate, you need target antigen, you need enzyme-linked antibodies, you need substrate solution. Now, what is the principle of ELISA? So in this, it is based on the antigen-antibody binding, enzyme reaction, and color change for the detection. There are different types of ELISA based on the binding, that which molecules are binding, what your sample contains, and what is present on the microtiter plate. So on the basis of that, there are different types. One is direct, one is indirect, then sandwich and competitive ELISA. Now, what are the application of this ELISA where you will see that this ELISA is being performed? So in different medical diagnosis, for example, HIV, pregnancy tests, or protein quantification or research, this ELISA is being used. Its advantages are it is having high specificity because antigen antibody binding is being observed and this binding is very specific. Then quantification, it will also help you in quantifying that what is the amount of antigen antibody binding which is being observed. Versatility, relatively simple and cost effective. And there are certain limitations, which means you need specific antibodies. And if you do not have the information about the specific antibodies, you won't be able to perform ELISA. And potential cross reactivity can also happen. Then limited dynamic range. So these are few limitations of ELISA. And in this figure, I have provided you the different types of ELISA. So when the antigen is bound to the microtiter plate and you are providing the antibody, so this will be the direct ELISA, and in which you provide the you have already bound substrate which is bound to the this antibody. You will provide the enzyme. If that substrate, if that binding will happen, then the enzyme will be able to catalyze this substrate and form the product, which will be in the form of change in color will be observed, due to which you'll be able to detect that whether binding has happened or not. So it entirely based on the positive binding. Greater the binding, more the color change you'll observe. If there will be no binding, then you won't be able to convert the substrate to the product and you won't observe any change in color. Similarly, in indirect ELISA, you are providing secondary antibody conjugate, which is containing the substrate, and then you are observing this color change. Similarly, in sandwich ELISA, you are already providing. So in this case, microtiter plate, it contains the antibody linked to it, and then you are providing the antigen which is bound to that antibody. And when your sample, you are entering your sample, it contains the antibodies which are specific to that antigen. So when it will bind, then you will provide the secondary antibody which is containing the substrate. And whenever the secondary antibody will bind to this antibody, then only this color change will be observed, otherwise color change won't be observed. So in this case, binding is happening between the two antibodies, secondary and primary antibody. But the first step is the binding of this primary antibody to the antigen. Then there could be the inhibitor antigen that could be present. So there will be the competition between them, that is known as competitive ELISA. Similarly, color change we will be observing, with which you can identify that how much fraction of your sample contains the antibodies or antigens and how much is binding. So let's move to next question, which says, what does Centeny refer to in comparative genomics? So you hear this word many times. So what does it mean in comparative genomics? Option 1 is the study of genetic diversity within a species, the study of conserved genes across species, the arrangement of genes on chromosomes in specific order, and the analysis of gene expression patterns. So what do you think? What does synteny means? Now let's see. So the correct answer is option number 3, which means the arrangement of genes on chromosomes in a specific order. So which is referred to as synteny. It is not the study of genetic diversity. It is not the study of conserved genes, because when you say conserved genes, so conserved genes means that uh, there could be some genes which are present in both the species. For example, when we say synteny, we means that they are present on the same chromosome. For example, there are three genes, gene 1, 2, and 3, which is present in chromosome 11 in mouse. Similarly, those 1, 2, 3 are present in the chromosome 10 in human, but they are present together. They are present in the same chromosome. So they will be the syntenic genes, which means they are present in the same chromosome in mouse also and in human also. So that means synteny is maintained. But when we say conserved genes, we say that the genes which are present in mouse, they are also present in human. So it is also possible that 1 and 2 are present in chromosome 11 in human and 3 is present in some other chromosome. So in that case, it is not referred to as synteny. Although those genes are conserved, but they are not conserved at the level of chromosomes. So when we say synteny, we are dealing with the information at the level of chromosomes and in the specific order. Now, the term synteny, it was introduced in 1971 by the scientist named John H. Renwick, and he defined synteny as the two genes residing on the same chromosome of the given species, even if genetic linkage cannot be demonstrated or tested. Synteny is derived from Greek word, which is one is syn, which means together, and tenia means ribbon, or can be freely translated as beads on the string. And the genes which are present on the same chromosome are referred to as syntenic, whereas genes present on different chromosomes they are referred to as asyntenic, and the state is referred to as asynteny. So in this, if you see carefully, the chromosome 11 of mouse and chromosome 8 of mouse, they contain certain genes, and these all the genes they are present in the same chromosome in human, that is chromosome 1. So if these three genes, these four genes which are present in the same chromosome in mus musculus, if they are present in the same chromosome in human also, then these genes will be the syntenic genes. Similarly, these all genes are also present in humans, so these genes will be the syntenic to these gene pairs. That means all these genes are present on the same chromosome, so that's why they are syntenic. Now let's move to the next question, which says comparative genomic analysis often involves the comparison of genomes of. So in comparative genomics, which genomes do we compare? Option 1 says multiple individuals within the same species. Option 2 says distinct species. Option 3 says only humans and primates. And option 4 says single-celled organisms only. So let's see the correct answer. The correct answer is distinct species, because we have discussed it a lot that in comparative genomic analysis we often involve the comparisons of distinct species. Although multiple individuals within same species can also be done, but when we say comparative genomics, we are oftenly studying different species. We are not limited to just single type of species or just closely related species, but we are often dealing with the distinct species. So don't get confused here. There are other options which might appear true, but we'll select the option which is more accurate than other. Now let's move to the next question, which says which of the following is not a benefit of comparative genomic analysis? Option 1 is identifying conserved genes across species, discovering potential disease causing mutations, determining the precise age of a species, and fourth is studying the gene regulation and expression. So, what do you think? Which cannot be the benefit of comparative genomic analysis? Because till now we have read a lot about comparative genomic analysis. We have seen its applications, how it is performed. So, by this time, you'll be able to distinguish between the options. You'll be able to get the correct answer out of it. Now, let's see. So, the correct answer is option number 3, which is determining the precise age of a species. So, this thing you cannot do with the comparative genomic analysis. As comparative genomic analysis, it focuses on the genetic comparisons and not typically used to determine the exact age of a species. And its benefits include identifying conserved genes, discovering potential disease causing mutations, and studying gene regulations. So, now quickly move to question number 18, which says genomic analysis has shown that closely related species tend to have. So, when we were talking about the phylogenetic analysis, then also we were talking about this thing. So now let's see what are the options. Option 1 is highly divergent genomes. Option 2 is completely identical genomes. Option 3 is some degree of genetic similarity and shared ancestral genes. And option 4 is no genetic similarities. So what do you think? The species which are closely related, what property they should show? So let's see the correct option. The correct option is some degree of genetic similarity and share ancestral genes. Now, many of you will be saying that why not completely identical genomes and if they are closely related. So, if you observe the species with completely identical genomes, yeah, it is possible that they will be closely related. But if you see, not many which are closely related will show this thing, this completely identical genomes. And then there are some means not closely related species which may show completely identical genomes, which are known as the paralogous genes, which are not closely related but still they show the identical genomes. Then highly divergent genomes. So this also we know that the two closely related species they show some kind of similarity. They are not entirely divergent. If they are divergent, they are not closely related. And then no genetic similarity, this is also not true because they share some kind of genetic similarity, some degree of genetic similarity, and they have same shared ancestral gene. So that's why this option 3 was correct option. Let's move to question number 19, which says the study of ancient DNA extracted from the preserved specimens, such as fossils or mummies, is known as. So, when you study the ancient DNA, then what does that study known as? Option 1, comparative genomics. Option 2, paleogenomics. Option 3, population genetics. And option 4 is phylogenetics. So, what do you think? Which is the study? So by this time, you know what is comparative genomics, you know what is phylogenetics. So among population genetics and paleogenomics, you have a brief idea about population genetics as well. And I'm not sure whether you know about paleogenomics or not. So now let's see. So the correct option is paleogenomics. So one thing which I want to tell you that there are certain questions in which you might know the correct answer, but in that case you can use the principle of being deselecting or removing the incorrect options. So you know comparative genomics, you know very well what does it undergoes, what does it means. So in that case you can means deselect or you can cut the options which you know that this won't be true for this. So at last you will be left with the option which is true. So for this you do not need to know about paleogenomics. You can simply cut down all other options which you know and then it will be left. So which means it is the study of ancient DNA. Now I have given here the complete detail of this paleogenomics, and this involves the study of ancient DNA, which are generally extracted from the preserved specimens. And these specimens could be fossils, mummies, or ancient remains. So, when I was talking about the phylogenetics, then when I was saying that these are the hypotheses, these are not facts. So, why? Because there are many fossils, many ancient remains which are present, which for which we do not know the sequence level information, which we do not have. So, as the data is being studied, we are now understanding, we are now getting more and more information due to which these phylogenetic trees might get changed, their arrangement can change, due to which they are just hypotheses. Now, what is the purpose of this paleogenomics? So, to understand the genetic makeup of extinct species, to investigate the evolutionary history, understand the relatedness between those ancient and the modern organisms, we can also reconstruct the ancient genomes and the genomes of ancestral human populations. So, these are all are the purposes why we perform paleogenomics. Now, what are the methods? How you can perform this? So, you can extract the DNA from those well preserved specimens. You can perform high throughput sequencing. You can take the sample, you can perform PCR to amplify that, and then you can perform next generation sequencing. And at last, you can computationally analyze those samples, you can reconstruct the genome, and you can compare them. Now, there are many challenges which you will face while performing these paleogenomics. So, those challenges are DNA degradation over time. So, with time, our DNA get degraded. So, if that remain will be very, very ancient, it is not well preserved, then it will be degraded. And then there are contaminations from the modern DNA, which is possible. Means if the fossil is being contaminated with the modern DNA, that is also true. Then limited availability of the well-preserved specimens, because there are very limited samples which are well preserved. Then fragmented and damaged DNA. So these are also the challenges which we face during the paleogenomics. Now, what are the applications of this paleogenomics? So, in that, you can reconstruct the extinct species genomes and then you can perform comparative genomics. You will understand, you can gain insights that what are the differences, how the humans have evolved, what is the migration pattern that was present. You can also study the ancient diseases, pathogens. You can also get the environmental history and paleoecology research. And the significance is that it provides you the window into the past. It will unravel the mysteries of extinct species. It will also give you the idea. For example, the fossils of dinosaurs, when the scientists studied them, they were able to know that when they could date them, that at what time those dinosaurs were present, how they got extinct, what were the reasons. So, all those things are only possible because we are just studying the fossils, we are understanding them, we are performing paleogenomics on them. So, it is providing us the insights into the genetic adaptations of ancient humans and their interaction with other species. It is not just limited to humans, it can also be expanded to other species as well. It will also help us to know what were the adaptations that they gained, what were the things that were missing which led to their extinction. And we can also reconstruct some extinct species by getting this information. And now there are certain notable achievements which this paleogenomics has got. So one such achievement is the complete Neanderthal and Denisovian genome reconstructions. So now these genomes have been reconstructed, and then understanding human migration patterns and interbreeding with the archaic hominins. So these all things, they were the achievements of this paleogenomics. So these informations now we have. Here I've provided you the brief flowchart that how it is performed. So you get the sample, which could be the some preserved sample, it could be the tissue, and it can tell you some means it could have some age as it is conserved. You will perform its grinding to obtain the DNA. You will get the extraction. You will extract the DNA. Now you also have to perform purification because there are certain DNA which will be degraded, or there could be some contamination that is present. So purification is done. Then the library is constructed. So for the DNA library construction, you can perform PCRs and you can construct a library. Then you will perform sequencing. It could be um, next generation sequencing. Different any method you can use of next generation sequencing based on the read size and depending on your results which you want to produce. Then mapping is done with the genome. So there is genome reference analysis which is being performed. Then you authenticate that, you observe the damage patterns, and you look for the contaminations. You will perform comparative genomic analysis. You will check that whether some microbial contamination or some other genome contamination is available so that you can distinguish the ancient genome with the current genome or modern genome. And at last, you'll perform genetic analysis, which will also tell you the demographic history, the phylogeny, selection signatures, and all these things you can infer with the help of paleogenomics. So I hope now you'll be aware of this term and you know how it is performed and what are the applications and what are the achievements of this paleogenomics. So let's move to the next question, which is horizontal gene transfer, a process where genes are transferred between different species, can complicate the interpretation of evolutionary relationships based on the genomic data. Which of the following is a common mechanism for the horizontal gene transfer? Option 1, sexual reproduction. Option 2, gene duplication. Option 3, viral vectors. And option 4 is mutation. So, we have talked about this horizontal gene transfer when we were talking about the xenologues, in which we said they had common ancestor, but now they have some genes which are transferred as a result of horizontal gene transfer. So, now you have to tell what could be the common mechanism with which this horizontal gene transfer has occurred. So, these are few options. What do you think? Now let's see what is the correct answer. So the correct answer is the viral vectors. When we say sexual reproduction, so whenever some gene component is passed on to the progeny with the sexual reproduction, it is the vertical gene transfer. It is not referred to as horizontal gene transfer. Then gene duplication. So gene duplication leads to the formation of paralogs and it is not some transfer. It is the duplication of the gene that is already present. So in this way, this is also not true. Then mutation. So mutation is also not the transfer. It means there is some change in the sequence of DNA which is already present in the species. But viral vectors, so vectors are the vehicles. That means the viruses, they are acting as a vehicle which is carrying some gene from the host and taking it to some the target. So in that case, viral vectors are acting as a source or the vehicle which is generally doing this transfer horizontally. Now in this, I have shown you the example. For example, here you have the bacteria E. coli, which has this antibiotic resistance gene. Now some virus infects it, it takes it's this particular gene, antibiotic resistance, and this horizontal transfer has occurred to this particular bacteria in which this gene is transferred to the bacteria Streptococcus pneumoniae, and this gene is coming from E. coli, so this gene will be the horizontal transfer gene. And it could be there could be other sources as well. This could be passed through the conjugation in case of bacteria, but in the other species, the viruses are playing the major role in transferring this. Here you can see there are different genetic components which accumulates in our nucleus, which adds to our genome, and they are occurring with the horizontal gene transfer. So, what are the different horizontal gene transfer which can occur? So, one is when from mitochondria some DNA get integrated to our nuclear DNA or the chromosomal DNA. So that will be a horizontal gene transfer. Or for example, from chloroplasm DNA is integrating to the nuclear DNA, that will also be a horizontal gene transfer. Or some endosymbiont is bringing some DNA which is getting integrated. That will also be the example of horizontal gene transfer. Or some viral vectors are bringing some DNA which is getting integrated to our nuclear DNA. That will be the example of horizontal gene transfer. Similarly, some interdomain conjugation is bringing some DNA, or some ingested prokaryotes or eukaryotes, they are bringing some DNA. So all these are the examples of horizontal gene transfers. And the mechanism could be the viral vectors, conjugation, or these mitochondria, chloroplast, endosymbionts, which are already present in the cell, and then they are leading to the integration of their DNA to the nuclear DNA. So all these things comes under horizontal gene transfer. So I hope now this concept of horizontal gene transfer is clear to you. So let's see the last question, which is question number 21, which says, what term describes the phenomena where two species that share a common ancestor evolve independently and develop differences over time? Option 1, convergence. Option 2, divergence. Option 3, speciation. And option 4, homology. So I hope, as we have discussed all those terms before, so I hope now you'll be able to answer this very quickly, because they are saying that the two species share the common ancestor. They have common ancestor, but they have now evolved independently and they have differences. What do you think? What is possible? Okay, let's see the correct option. So the correct option is divergence. So divergence means when the two species they evolve independently and accumulate differences due to which now they have diverged. So divergence only happens when something is beginning from the same point and then it diverges. But when we say convergence, it means there are two separate points which are converging to a single point. So it means they will be having the different ancestral origins. So in that case, it will be the example of the um, analogs. Whereas in divergence, it is the example of the orthologs, which have the same ancestor, but now they have diverged in their level of functionality, or at the sequence level, they have accommodated some differences. Okay, now I am giving you few differences between the convergent and divergent evolution. So the convergent evolution is the process where the distantly related species develop the similar structures as the adaptations to the environment. When we say divergent evolution, it means that interbreeding species, that means which were previously were the coming from the common ancestor, they diverge into two or more descendant species. So now they are split into, there is a speciation event which is happening over here. Now both species live within same environment in the case of convergent evolution, whereas the two species they live in different environments. As they are living in different environment, they will accommodate different changes due to which they are showing these differences. Then convergent evolution is seen in the unrelated organisms evolve similarity while adapting to a common environment. So as environment is common, so they are having the adaptations to the same environment due to which they appear as if they are evolving from the same ancestor, but although they have different ancestor. Whereas in divergence, as two different species are now present, and it results in the two species becoming less like the common ancestor, due to which it appears as if they have diverged from the common ancestor. It occurs through the developing homologous structures, and it occurs through developing the analogous structures in the divergent evolution. Examples include ostriches, rheas, and emus. And in this, examples include the dinosaurs, Darwin finches, four-limbed structures of the vertebrates. So, in divergent evolution, if you see, there is a wolf, which is the common ancestor. Fox and domestic dog are arising from it. Although its ancestor is same, but they both appear a bit different from one another. When we say convergent evolution, we see that there is no winged common ancestor which was present, but birds, butterflies, bat all have wings, and it is helping them to fly. So it appears as if these wings are coming from some common ancestor, but there is no common ancestor. So it is known as convergent evolution, in which the ancestor is not common, but they all are converging to a same function, which is their wings. So I hope now you are aware with these terms. One is convergent and one is divergent. So now there are different doubts which you have asked me. So in this, I'm trying to resolve the doubts. So the first doubt was regarding the syntenic regions, in which you have asked that uh, what does syntenic region means, and then you have also asked that in which organisms you will see the syntenic regions. When do you say that the particular region is syntenic? So syntenic regions are the portions in the genome in which the order and orientation of genes or genetic elements are conserved across different individual or species. So this syntenic region, you will only be able to define it when you are comparing it. So it is the relative term, which depends. For example, if you are comparing human and mouse, then if the genes which are present in the mouse and if those genes are present in the same chromosome in human as in the mouse, then you will call it as syntenic regions. Whereas there could be some genes in the zebrafish which are present on different chromosomes which are present in the same mouse chromosome. So in that case, if you compare human and zebrafish, then also you can identify the syntenic regions. So in this way, the definition of syntenic regions or the genes which are occurring in the syntenic pair, they can vary if you select different individuals for the comparison. Whereas there is a broader term as well. So these syntenic regions can also be defined by comparing multiple species. And by comparing multiple species, you are saying that which of the genes which are in the same chromosome in all those species. So in that also you can define the syntenic region. So synteny is often used to describe the conservation of genes, gene order across the genomes. Now there are certain key points which you should always keep in mind. So you also asked me that what is the impact of these syntenic regions in the genome organization. So these why these syntenic regions are important. So it helps in the conservation of gene order. It indicates that these, as the same genes are present in the particular order and orientation in the genome of different organisms, and this conservation of gene order can extend over the varying distances along the chromosomes. So there is this conservation of gene order. These genes are remain conserved with the help of the syntenic regions. Now, what is the impact on the genome organizations? Functional clustering, which means that these syntenic regions, they often contain functionally related genes or the genes which are helping one another in their functions, or they are sharing some common pathway due to which if they will be present together, they will be expressed together and they can coordinate their function. So this functional clustering is one of the impact on genome organization. Then evolutionary insights. So comparing the syntenic regions across species, it can also provide you the evolutionary information that in different species, if some genes are still present on the same chromosome, then it will tell you that this has some role to do. And it also help you to study the gene duplications, their rearrangements, and their evolutionary events. Then genomic evolution. So these changes in the syntenic regions will also tell you that if particular gene is changing its syntenic region, that means now it is evolving its function or it is having some kind of effect. It is evolving. So in this way, by comparing these syntenic regions, you can also get the information at the level of evolution. So it's with the evolution, it is also impacting the genome organization because this change such as gene duplications, inversions, or translocations, it can drive the genomic evolution and contribute to diversification of species. Now, if to describe that where you can observe the syntenic regions, either it is only observed in animals or in plants. So there are different organisms in which the syntenic regions are common. So such as animals, plants, and microorganisms. And in plants, especially in plants also, the syntenic regions are observed, and not just plants and animals, but in microorganisms also you can find the syntenic regions. Now, degree of conservation. So these syntenic regions, their conservation can vary widely among different species. And when you say that this region is highly conserved across distant species, so the species which are very distant, this region is highly conserved, while the others may be conserved only among closely related organisms. And then genome analysis. So this synteny information can also help you in genome assembly, annotation, and comparative genomics. So when you are comparing multiple species, then it can help you to assemble the genome. It will also help you to annotate the genome because now you know that these two individuals are very closely related. So there are more chances that their genes will also be present on the same chromosome. So it will help you in annotating the functionality or the annotating the genes at the genome or assembling that genome. So these are few advantages of the syntenic regions. And examples of these syntenic regions is the human, mice, fruit flies, plants like Arabidopsis thaliana, or even bacteria like Escherichia coli. They all contain the syntenic regions. So now the next doubt was regarding the expression, in which you asked what is the difference between the genomic expression, gene expression, and RNA expression. So when we say genomic expression, we talk about all the genes and the known coding elements. So in this, we are talking about the gene regulation and the activity across the entire genome. Whereas when we say gene expression, we are focusing only on the information or only on the DNA sequence, which is used to synthesize some functional gene products, typically proteins. So all the genes which are forming the protein, their expression, when we check their expression or their functional gene products, that is known as gene expression. Whereas in genomic expression, the gene which is forming all the mRNA as well as some known coding elements, that is also referred to as genomic expression. But when we say RNA expression, it means the level of various RNA molecules such as mRNA, non-coding RNA, ribosomal RNA in the biological sample. So this is known as RNA expression. Now, what is the scope? So it involves all the genomic elements, including protein coding genes, known coding genes, pseudogenes, and more. So these all the genes, they will be under the genomic expression category. Whereas if in gene expression, we are only focusing on the individual genes and their transcription into the RNA and those genes which are forming the proteins in the end. When we say RNA expression, it encompasses all the RNA transcripts, both coding and non-coding in the transcriptome. So in this, you are observing the transcriptome in which you are focusing on all the RNA transcripts. And means it could code for the protein, it could not code for the protein. Then comes measurement methods, how to measure the genomic expression. So there are genome-wide techniques such as whole genome sequencing and epigenomic analysis, which you can perform for genomic expression analysis. Then for gene expression analysis, you can perform RT-PCR and RNA-seq or microarrays or northern blotting to quantify the specific gene transcripts. Whereas in RNA expression, you can perform RNA-seq, qPCR, which is the quantitative PCR, or in situ hybridization to measure the RNA abundance and identify the RNA species which are present in the sample. Now, information which it provides is that it provides you the insight into the global gene regulation, the known coding RNA functions, epigenetic modifications, and genome-wide patterns of gene activity. When we say gene expression, it provides us information about the transcriptional activity of specific genes, including the mRNA levels and alternative splicing. Because we are only dealing with the mRNA in the gene expression, which are synthesizing proteins. So now if we talk about RNA expression, this offers us the data on the abundance, localization, and types of RNA molecules which are present in the given biological context. Now, what are the application of genomic expressions? So it will help you to study the genome-wide gene regulation, comparative genomics, epigenetics, and functional role of known coding elements. Whereas the gene expression can help you to investigate how genes are regulated, what are their roles in biological processes, and how they respond to disease or stimuli. So all those things you can use by observing the gene expression. You can alter certain things and you can see that how this gene expression is varying. So in this way, you can get the inference, you can get the results that this particular change is leading to a change in the gene expression. Then in RNA expression, it provides insights into the cellular processes. It will tell you the means at what stage the development and what developmental phase a particular organism is, because RNA expression is also varied at the level of age, or it can vary at the level of tissues. There are different transcripts which are expressed in particular tissue and not in the other tissue. So this can provide you the insights into the cellular processes, development, disease mechanisms, and role of specific RNA species. For example, microRNAs, long non-coding RNAs. Now, examples of genomic expression is the comparative genomics, epigenetic studies, and identification of non-coding elements. Whereas gene expression example is the gene expression profiling and validation of specific gene targets, analysis of regulatory mechanisms. And the example of RNA expression is the transcriptome profiling, quantification of RNA species, and spatial analysis of RNA distribution. So all are the examples of RNA expression. Now there was another doubt in which you asked about the different methods which we can use to analyze the expression. So we can use reverse transcription PCR, that is RT-PCR. So when you want to see the expression, so we can use RT-PCR, which help us to quantify specific RNA molecules by reverse transcribing RNA into cDNA and then amplifying it using the PCR. And its application is measuring the expression of selected genes, validation of high throughput data. Then another method is RNA-seq, which is known as RNA sequencing. It sequences the cDNA synthesized from the RNA samples to profile the entire transcriptome. Now, transcriptome profiling, quantification of gene expression, and detection of alternative splicing are some of the applications of RNA-seq. Then you can also use microarrays, which uses the immobilized DNA probes to measure the expression of thousands of genes simultaneously. And its application is large-scale gene expression profiling and discovery of differentially expressed genes. Then you can also perform Northern blotting, which helps you to detect and quantify specific RNA molecules through electrophoresis and hybridization with the labeled probes. And its application is in the RNA size determination and quantification of specific RNA transcripts. Then you can also perform in situ hybridization. So you might have heard the term fluorescent in situ hybridization. So it is the one the same because in that you use the fluorescent labels. So visualizes the spatial distribution of specific RNA molecules within the cells or tissues using the labeled RNA probe. So in this you label the RNA probes and you look for their distribution. So application is studying the tissue specific gene expression patterns and identifying the cell types expressing specific genes. So these are application of this in situ hybridization. Then when we say quantitative PCR arrays, so it measures the expression of multiple genes simultaneously in the predefined sets using the real-time PCR. And it helps you to study the predefined gene panels, pathway analysis, and biomarker discovery. Then there is another method which is single cell RNA sequencing. It profiles gene expression at the single cell level by sequencing cDNA from individual cells. So at particular time you are only dealing with the single cell. So in this case, you are studying the cellular heterogeneity and developmental biology and disease research. So when you are targeting a single cell, then these are the different applications of this single cell RNA sequencing. So these are different methods and these are their principles and typical applications. So by now, you'll be able to know that what is your aim. And by looking at your aim and the data you have, you can select the method which will help you to cater your aim. Now, the another doubt was about the in vitro translation, in which the few students have asked that what is the difference between the in vitro translation from in vivo and how it is performed. So, in vitro translation means translation is the conversion of RNA to proteins. In vitro means it is performed outside of the cell in the in vitro conditions, in the lab conditions inside the test tube or reaction vessels. It is the cell-free synthesis of proteins. So for this, we require similar things as we require in the in vivo, such as you require mRNA template, you also require ribosomes, you require amino acids, which are attached to the tRNA. Then you require energy source because it is the energy consuming process such as ATP and GTP. You require enzymes which will catalyze these reactions. You require buffers and salts to maintain proper reaction conditions which are maintained in vivo. The cell is maintaining it by itself, but in external cases, you require the buffers and salts to replicate the same conditions as the cellular conditions. And then comes the factors which decide that whether this translation will be efficient or not. So mRNA quality. So this purity and integrity of mRNA template is very important to ensure the translation will be accurate. Then ribosome activity. So the functionality of ribosomes is also very important, which will decide the efficiency of the in vitro translation. Then amino acid availability. So you also require the amino acids that should be present on the tRNA anticodon, which should be complementary to these mRNA codons. So if they will not be available, then how this translation will proceed? So it means amino acid availability is also one of the important factor which govern the efficiency of in vitro translation. Then comes reaction conditions. So you have to maintain pH, temperature, and salt concentrations because if you disturb them, then it can affect the efficiency of this in vitro translation. Then presence of inhibitors. So there are certain contaminants or substances which can interfere with this translation process and it can reduce the efficiency. So all those factors you must check to ensure the efficiency of this in vitro translation. Now, if we compare in vitro and in vivo, so we'll observe that there is the lower efficiency of these in vitro translations when we compare it with the in vivo, because in living cells, we have proper conditions, but it's very difficult to maintain all the cellular machinery and regulatory mechanisms outside the in vivo, means outside the cell to provide same conditions. It's a bit difficult due to which efficiency is lower in the in vitro translation. Now, what are the applications? Why do we perform in vitro translation? in vivo translation is better than in vitro. So we use this for the protein production. If we want to synthesize specific proteins for research, diagnosis, or biotechnological applications, so we use this in vitro translation. Or we can also perform protein labeling, which means that we can incorporate the labeled amino acids to allow the production of radio-labeled or fluorescently tagged proteins for assays. So we perform different assays for which we require the proteins. So to perform that, we need in vitro translation. Then protein folding studies. We can also study the protein folding, misfolding, and aggregation in the controlled environments. Because if we perform it inside the host, then it will have some consequences. So due to which, to perform these experiments externally, we require to synthesize these proteins in vitro. Then functional assays can also be performed by in vitro synthesis of proteins. Then in drug discovery, so these drugs can also be synthesized by performing in vitro translation. Vaccine development, so for the production of antigens for the vaccines, we can perform them in vitro. Then ribosome research, so studying the ribosome structure and function in the simplified system. So in vitro conditions provide us the simplified system in which we can perform these studies, because in vivo it's difficult to study the particular entities such as ribosomes. Now there was one of the doubt regarding the transposons, in which you asked that are they code for some particular protein? What is the role of transposons? Are they just the repeat sequences? Because when we say transposons, we call them as the defunct regions of the genome. We call them as the known functional part. We call them as the repeats. So this is the review article which was published in the Nature Cell Biology. The people who are interested or who want to read it, they can go through this. So this was the review article in which they have compared the mammalian genomes, and they have checked that how these transposons they have led to the evolution in the genome, how they caused the innovations, how it is working on the mammalian genome. So in this they look for the how the host genome sequences introduced by the transposon activities provide the raw material for genome innovation, and they have also documented the distinct evolutionary path of each species. So in this they are saying that these transposable elements, there are co-opted proteins, there are chromatin boundaries, they can act as enhancers, promoters, they affect splicing, they can act as the poly A tail, which can determine the sex. They can provide co-opted RNA, which can help in the cancer resistance or chromatin accessibility. So there are multiple role of these transposable elements. They can influence the expression of genes. They can itself code for some other genes. So now let's uh, look at them in more detail. So in this paper only, they have mentioned, they have identified that these transposon-derived gene regulatory elements, they diversify host gene isoforms and enrich the expression regulation modality. So in this figure, you can see that these boxes in the pink rectangles, they represent the transposable elements. And in this, these transposable derived sequences, they contribute to gene enhancers. So now these transposons can also contribute to the enhancers, promoter regions, exons, terminators, splicing acceptors or donor sites, chromatin boundaries, or regulating the structure and expression of proximal host gene isoforms. So these are the roles of these transposons. So we cannot say that these transposons are just the repeats and they are means randomly present. No, we cannot say that because they have very specific roles and very important roles. So these are some of the functions of the transposons, some of the contributions made by the transposon derived sequences, and their domestication expands the gene regulatory modality. So domestication means their expression simply. So it helps in expanding the gene regulatory modality. It can enrich the transcript diversity. It can also improve the functional reservoirs which are present with us. It can increase the functionality, and it can lead to multiple functional transcripts that could be produced. So in these figures, the pink rectangles represent the transposable elements, and these blue rectangles represent the protein coding exons, which are normally present. So now this transposable element, it is containing some promoter region or enhancer region, and now it is present on the promoter. So transcription factors will bind to it, and it is causing the expression. So in this, you see that in this case where this transposable element was not present in this transcript, so in that case the protein was formed like this. But when this transposable element is present in this type of transcript, so it is forming the proteins like this, which is containing some altered sequence. So in this way, you will know that now this gene can form two different transcripts with different sequences. So this sequence variation can also have some functional implications. Maybe these two proteins will perform some different functions now. Maybe this transposable element is contributing to some domain, functional domain, or it may have some regulatory role. So these are the a promoter when the transposable elements are acting as a promoter. Now, if they are acting as enhancers, so they will allow the transcription factors to bind and they will regulate the expression. For example, previously when its expression was only the four proteins that were expressing, so now multiple proteins are expressing. So it is enhancing the expression of this particular mRNA. Now, if it is acting as an exon, so in that case, in one of the transcript, this particular exon is coming. So now it will lead to the formation of the protein product, which will contain this sequence from the transposable elements as well. Whereas in this case, this transposable element is not coming in the transcript, so the protein is different. So similarly, in the terminator, when it is present in the terminator site, so in this way, the different transcripts are formed from the mRNA. Then it can also have its role in splicing, in which it will perform variable splicing. In this case, three exons are present, and in this case, only two exons are present. So this transposable element is lying in the intronic position, and in this case, it is performing the splicing in such a way that in one of the transcript, one exon is being skipped, which is present in the normal isoform. So in this case also, it is producing two different transcripts or two different protein products. Then the TAD boundaries. So similarly, in this case also it can act as a regulatory element. So this star represents the gene regulatory element and this hexagon represents the CTCF. So in this case also these transposable elements are having different roles in defining the boundary between these two elements, gene 1 and gene 2, whereas if it was not present, so there is no boundary between both of them. Now, the next thing which they discussed in their paper was that these transposons they also confer unique modes of cis gene regulation in the host genomes. So, in this case, this is the species-specific gene regulation in which these transposable elements have different regulatory mechanisms. For example, in mouse, if you see, it is enhancing the expression. So, multiple transcripts are expressed, and at end, multiple proteins will be formed. Whereas in human, it is not having that much role due to which only limited number of transcripts are being formed. Whereas if you see in the pig, it is not causing due to mutation in this transposable element, it is now this gene is not able to express itself. So in this case, it is a species-specific gene regulation which occurs when distinct transposable elements integrate proximal to homologous genes across species, generating a unique expression pattern. So these genes are shared among all of them. They are common ancestral genes, but their expression is variable among all these three species. And this variable expression is only caused with the help of these transposable elements. Now, if you see the middle box, it represents the convergent gene regulation in which these are different species and these are different transposable elements. But if you see this particular gene coming from similar ancestor, it is expressed in the similar level, which means that convergent gene regulation means when distinct transposable element insertions across species converge on the same regulatory principle to yield similar or identical expression. So these transposable elements, they are not altering the expression, their regulation is convergent. It's similar in all. So why it is convergent? Because these transposable elements are different, but still they are performing similar regulation. Then is the coordinated gene regulation. So coordinated gene regulation means a similar transposable element is present in the different genes, but their expression is same. There is the coordination between their regulation. So coordinated gene regulation occurs when the related transposon elements from the same transposable element family spread in a given host genome and land proximal to cohort of host genes to coordinate their expression. So these are different ways in which transposable elements is affecting the regulation of genes. These genes could be common coming from the common ancestor or maybe different. And the another thing which they have mentioned is the co-option of the transposon encoded proteins contributes to new host biology. So when we say transposons, so they can themselves code for different proteins. So for example, here I've talked about the retrovirus which is containing the LTR, long terminal repeats, and these are the genes. So now this virus is bringing this particular genetic material, and it will get integrated into the host genome. Firstly, there will be membrane fusion in which this virus will fuse with the genome, with the membrane. Then there will be uncoating. This protein coat will be removed, and these genetic elements will be exposed. There will be reverse transcription, and then there will be transcription. After reverse transcription, these mRNAs will be, because it is representing the retrotransposons, which means they are coming from the DNA element after copying to the RNA intermediate. So this RNA intermediate, when it enters the host, it has to be converted to the DNA with the help of reverse transcription. This particular DNA get integrated into the nuclear genome, and when it integrates to nuclear genome, when the nuclear genome will be expressed, this particular part will also be expressed, it will be transcribed, and after transcription, it will be spliced, and then it will enter the cytoplasm where it will be translated. It will convert to few proteins, which will further help to synthesize the proteins and the coat of this particular retrovirus. And this, there will be assembly and packaging, and then budding will happen, in which this virus will again be formed. So in this way, integration is also done and virus molecule is also synthesized. So there are different genes which have different pathways, which can have different roles. So this diagram illustrates the same thing. So in this bottom diagram, you will see these are the examples of domesticated GAG and the ENV genes. So these are GAG and ENV genes, which are repurposed for the neuronal functions, host defense, and placental formation or placenta development. And the remarkable modern innovations they conferred by the retrotransposon encoded proteins, it can be traced back to their proviral functions. So they have certain functions. They can provide you with the protection. They can also be helpful in the repurposing neuronal functions. So these are few of the functionality. And to answer your question, which you asked, that either these code for proteins. So yes, some of the retrotransposons they can code for the protein also. Means they are not entirely defunctional. They are not just the repeats. They contain some of the region which can code for some proteins. Now in this, this was related to the doubt which you asked about the chipsec, that what are the application of chipsec and can it replace the other methods which are available such as RNA-seq or some other methods. So these are the applications where we use the chipsec, such as identification of the binding sites or functional annotation of genomes or transcription factor analysis, epigenetic studies, identification of enhancers and silencers, comparative genomics, cancer research, drug discovery, functional genomics, personalized medicine, validation of computational predictions, disease mechanism investigations. So these are different applications. You can go through this table and you'll be able to understand what is chipsec. But when we say chipsec, we are dealing with the sequence of the DNA which is binding, which is interacting with the proteins. And those proteins can be the transcription factors or the histones. So when we are identifying those genomic regions which are performing this binding, then we are saying it is the application of chipsec. But when you are only want to sequence the genome, when your purpose is to sequence the genome, then you don't go for chipsec, because chipsec is having its role in the protein and DNA binding interactions, not in the sequencing. So this is the few applications, and you cannot say that chipsec can replace RNA-seq because function of both chipsec and RNA-seq are entirely different. So these are few of the doubts which you have in this session. So I hope I have cleared most of your doubts. But till now, even if you feel that there are certain doubts which you want to clear, so you can, if you are watching this video, then you can comment in the end and you can write down your doubts, and I'll be happy to help you with your doubts. I'll give your answers. So now this was the last session related to this functional genomics course. So I wish you a very good luck for your exams, and I hope that you have learned so many things from this functional genomics, and I am also very happy that I have also learned many things from you also, from your doubts also. So thank you so much, and very good luck for your life.